Hello, good evening. We are in the last topic of the molecular genetics section of your course, SIL 1001. And this involves the topic of cloning or recombinant DNA technology. Some books call it genetic engineering, genetic manipulation. There are various names for it, but basically they do the same thing. So I'm going to go through the slides and you'll see me jumping here and there because the slides are not really ordered nicely. So never mind, just follow through. And again, as usual, if you have any questions, please let me know. Okay. Now, basically in cloning, right? It's as a process in biotechnology where you use bacteria to produce a protein of desire. All right. Let's say, for example, you know the protein insulin, right? Insulin, well, it's one of the first proteins to be induced by biotechnology. What they did was they take the human gene, the insulin gene, and put it in a bacteria so that bacteria can produce insulin in bulk. So that is in theory why they do it. And if you look at the detailed process, you'll find that this is the DNA fragment to be cloned. We're going to look at this later. This is a DNA fragment. And then this is a cloning vector. Every time you want to do cloning, you must have a vehicle, right? You can put it in the bacterial DNA, but it's going to be slow. I'll tell you why. But basically, you in this case, using plasmid, you put it in a plasmid, and then you create a hybrid, and this is what we call as recombinant DNA. Recombinant comes from the word recombine, so that means you have this in this picture. You have this green fragment. You join it with the yellow, so you have a mixture, a hybrid, and this is called recombinant DNA. And then you put it back into the host cell. So in this case, the host cell is a bacteria. The bacteria will read that DNA as part of the expression process and produce the proteins that you want. All right. So this is in principle. Well, of course, it's very direct. It's very straightforward. But we are going to look at the process so that you'll be able to understand the requirements a little bit more. Now, basically, again, there are two reasons why people clone, right? The putting in genes in bacteria. There are two reasons to it. One is if you want something to be produced in bulk, you have one copy, you put it in bacteria. We know bacteria will grow and divide and propagate. If you put your DNA fragment inside the bacteria properly, the correct way, the bacteria will make copies of it. As it makes copies of itself, it will make that DNA copy for you. So basically, you take the fragment that you want, put it in a vector, you put it in a bacteria, and just let it grow. And once the bacteria grows more and more into a bigger culture, then you can get the fragment out again, right? So essentially, you'll have a lot more of that. So you clone because you want to produce in bulk, define small pieces of DNA. The reason that pay attention to small. You're not talking about trying to produce the whole human genome. So a particular gene you can put in bacteria so that it can produce for you. So one is making copies. The second reason why people clone is actually to produce, as I mentioned earlier, to produce particular proteins, and this is called heterologous protein expression of heterologous. Heterologous means hetero is a combination of different, homologous. Chromosomes are homologous. So heterologous means in that particular organism, it has a foreign DNA. So if you put it in a foreign piece of DNA and then you ask the bacteria to produce whatever that is coded by that piece of DNA, the protein that is produced, and which will be able to take it out later. And this is the whole process is called production of protein in a heterologous system. And this has been used in the fermentation industry. As I mentioned, insulin is one of the first biotechnological protein that's been produced in bacteria. They are used by pharmaceuticals, production of vaccines, and also for molecular diagnosis. You use that to produce antibodies or whatever gene so that you can use it for medical purposes. Right? So again, take home message from this slide. You clone, it says cross species. Cross species barrier means you can take a human gene and then put it in bacteria. Of course, there are certain rules that you have to understand, certain limitations, because why do I say that? Because bacteria, remember I mentioned about, somebody asked about exons and introns. Well, bacteria doesn't have exons and introns, so it doesn't have to do splicing. Human cells or eukaryotic cells, you have introns and exons. So there is a process called splicing, right, where you need to remove introns and then put in exons. So if you put in a fragment that contains exons and introns in bacteria, well, the bacteria will not be able to process it. So basically what you put inside bacteria are just the exons, right? You cannot, so you have to process it before you put it in the bacteria. Okay, so those are some of the considerations. Um, basically that will be the main consideration, because you have to make sure that the bacteria is able to produce the gene correctly. Um, we'll okay. This is the figure again. So okay, just to this is the explanation. If I can put the thing in center. So here you have, this is the cell containing the gene of interest. This is what you want. Like, let's say for example, I mentioned the insulin. So you get the DNA out. This is the bacteria. Now note that the bacteria has the bacterial chromosome, and then the bacteria also has plasmids. Now plasmids is also called extra chromosomal DNA. The bacteria can live without it. The bacteria can live without plasmids, but plasmids give the bacteria some additional abilities. There are various types of plasmids. Some give the bacteria antibiotic resistance. Some gave the bacteria toxicity. These are extra chromosomal. The bacteria for day-to-day life doesn't need that plasmid, but they are found as part of the bacterial evolution process. And then these are the molecules that have been used a lot in the process of cloning. So we take the plasmid out, we take the gene out, join them together, and then here you get your recombinant DNA, and then you put it back into the bacteria. As the bacteria grows and makes copies of itself, it also makes the copies of the plasmids for you. And then this is the one reason you want to get to do cloning is to make copies. So copies of the gene, so then you get the gene out again, and then you can do studies on it. And then here's an example of gene for pest resistance. This is studying the gene, even for bacteria for that sort of absorbs or clean up the oil spills. Or you can produce proteins, right? And then you can harvest the proteins and then use it for medical purposes. All right. We won't look at this now, perhaps later. So in the process of cloning, you will require, of course, you want to have something that you want to clone, the gene of interest. You want to have the transport vehicle. In this case, we are going to talk about plasmid. There are other types of transport. You have cosmids. You can put it in YAC, use artificial chromosome. There are various, this called transport vehicles, but in this case, we're going to talk about plasmids and also host cells. In this case, host cells are bacterial cells. Again, you can clone in yeasts. You can clone in mammalian cells. There are various cloning systems, but for the purpose of this course, we are going to talk about, we are limiting the study to bacterial systems. So transport vehicle, we're talking about plasmids. Host cells, we're talking about bacterial cells. All right. Now, in order for you to be able to do this, well, you've seen that this one you have the DNA fragment to be cloned and joined and so on. You see that bit. Here it is mentioned again, basic steps. You need to get the gene out. Where's the picture? Yeah, you need to you get the gene out. So you need you need to make recombinant DNA plasmids, and then you are able to put it in the bacteria. So all this you need tools. To do all this you need tools, and the main tools or aspects or processes that are involved in cloning are these ones. And these are the things that we, well, some of them again, we are going to look at a little bit closely so that you can understand the process of cloning in bacteria. So you, first of all, you must be able to extract DNA. You must learn how to get the DNA from out of the cell. As I said, as mentioned, in bacteria, you have the bacterial DNA, you have the plasmids, but you just want the plasmid, you don't want the bacterial DNA. So there must be methods to actually do it. And in the human genome, for example, you have about 3 billion bases, but you're actually looking for one particular gene. There must be a way to do that as well. So you need to isolate the fragment out. You need to isolate the plasmid. You need to separate DNA electrophoresis, right? We've mentioned that, I think. Oh no, we haven't. Electrophoresis is I'll show you later, but basically you're separating the DNA based on fragments. You need tools to cut DNA. You need tools to join DNA. We know that already. Joining DNA is, remember, in replication towards the end, right? You have polymerase synthesizing the fragment, and then towards the end you have the Okazaki fragments will be joined, and this is done by ligase, and this is called the process called ligation. There's a technique to amplify DNA because bacterial DNA, sometimes you have very low copy number, so you don't have that much. You need to make a lot more, and then you talk about cloning and PCR and processes. I mentioned that you build the recombinant DNA, and then you need to put it back into the bacteria, and that's the process is called transformation. And also, what's also important, because all this occurs in a tube, right? You don't actually see the fragments. When you want to create the recombinant DNA, you get a lot of plasmids, you get a lot of fragments, you put them together in a tube, and then you put in the enzyme, and then you wait, and then you must be able to take it out again. You can't see these things happening with your naked eye. Even using microscope, you cannot see the fragment being joined and so on. You know, but you just add these things. You add DNA plasmid, you add the fragment, and then you add the enzyme with buffers and so on. And then you take this out, and then you put it into the bacteria. It's a process called transformation. So again, you have lots of bacteria. Some of them will take it, some of them will not take it, but you will be able to choose or select which ones have taken up. And this is the whole process, what we call as selection. We're going to do that as well. So introduction purposes, you need to understand why you clone, what are the steps involved in cloning, and then what are the tools or processes that are involved so that it will help you out to the process of cloning. All right. So getting into specifics, I'll do that in the next video. I'll see you then.